Hi everyone, this month we're going to talk about the microbiome. And just as a reminder, all information presented and written in this video is intended for informational purposes only. You should not rely on this information as a substitute, nor does it replace professional medical advice, diagnosis, or treatment. If you have any concerns or questions about your health, you should always consult with a physician or another healthcare professional. What is the microbiome? Maybe you've heard some recent research on it, or you've heard it on commercials, maybe specifically a yogurt commercial saying that it helps with your gut microbiome. But what it is a collection of all microbiota that resides on or within human tissues and biofluids, along with the corresponding anatomical sites in which they reside, including the skin, the mammary glands, the placenta, seminal fluid, and the uterus and ovarian follicles, the lungs, saliva, and the gastrointestinal tract. Types of human microbiota include bacteria, archaea, fungi, and viruses. Humans are colonized by many microorganisms. The estimate in recent studies estimates that the ratio of human to microorganism cells is 10:1. Some microorganisms that colonize humans are commensal, meaning that they coexist without harming humans, and others have a mutualistic relationship with their human hosts. As you can see in this picture, there are multiple sites and types that reside in or on the body. It is estimated that the total weight of non-human cells is around 4 pounds of the total body weight. Microbes are extremely important. The more research goes on, the more we find that it impacts our health in a variety of ways. One of them is our metabolism. It helps synthesize specific vitamins such as K and B. And it's also really important, the bacteria in there is really important for helping synthesize and help process our essential amino acids. The gut-brain axis is a biochemical signaling that takes place between the gastrointestinal tract and the central nervous system. The gut flora can produce a range of neuroactive molecules such as melatonin and serotonin, which are essential for regulating sleep sensation in the gut and mood and sleep. Changes in the composition of gut flora due to diet, drugs, or disease correlates with the changes in levels of circulating cytokines, some of which can affect brain function. The gut flora also releases molecules that can directly activate the vagus nerve, which transmits information about the state of the intestines to the brain. It also affects our immune system, both the adaptive and the innate immune system. And it's also correlating to our genetics, specifically with epigenetics. Epigenetics is the study of how your behaviors and environment can cause changes that affect the way your genes work. Gene expression refers to how often or when proteins are created from the instructions within your genes. While genetic changes can alter the protein it is made, epigenetic changes affect the gene expression to turn genes on or off. Since your environment and behaviors, such as diet and exercise, can result in epigenetic changes, it is easy to see that the connection between your genes and the behaviors in the environment is very important. The first microbiome research took place in the late 1800s in Europe. Following the initial observations of intestinal microorganisms, German pediatrician Theodor Eschrich consolidated the study of the human gut flora. Later, his work was continued by Henry Tisler in Paris, who administered probiotic bacteria to children and adults to help improve their gastrointestinal conditions. In recent studies, a very, very important study that was done on the human microbiome. It was called the Human Microbiome Project. It was a 5-year-long international effort to categorize the microbial communities found in the human body and to identify each microorganism's role in health and disease. The project capitalized on decreasing costs of the whole genome sequencing technology, which allows organisms to be identified from samples without the need for culturing them in the laboratory, from technology also facilitating the process of comparing DNA sequences of microorganisms isolated from different parts of the human body and different people. It was estimated that they found over nearly 200 different bacterial species within all of their test subjects. So how do we get our microbiome? Well, it actually starts before we're even born. Developmental, it starts with the mother within the placenta and her microbiome diversity that she has. Also modes of birth. If you're born cesarean versus vaginally, that can affect the microbial differences. During infancy, there's a huge window here for development. There's factors such as being breastfed versus formula, geographic location, where you're born and where you grow up, specific family members, such as being exposed to siblings or cousins, host interactions, what kind of environment you're around, if you're living in an urban setting or more rural. Even your mother's diet while being breastfed can affect your microbiome, and weaning, what kind of food you get weaned off into. In this study in 2010, it shows the effects of the microbiome diversity versus being vaginally delivered or via C-section. As you can see here, there's certain types of strains that are different versus vaginally or C-section. Antibiotic use affects the microbiome greatly in infants and children. A growing number of studies have shown that antibiotics can result in microbial dysbiosis and the disruption of the gut microbiome in neonates and adults, contributing to numerous diseases, including diabetes, obesity, inflammatory bowel disease, asthma, rheumatoid arthritis, depression, autism, and superinfections due to antibiotic resistance. There was another study done between 2006 and 2013, which observed inflammatory bowel disease issues with children under 6 years of age. And they found that antibiotics during pregnancy significantly increased issues within the gastrointestinal of children under 6. There's a lot of factors that can affect your microbiome. Like I said before, being breastfed versus formula fed. Types of diet is a huge one. A meat diet versus a vegetarian diet is very important. Being around pets, since they are outside, they can increase your microbial diversity. City living, as I mentioned before, air pollution can also affect your microbiome. Culture, so where you grew up, food habits, certain rituals. Geographic location, as I mentioned before. Genetics, which refers to the epigenetics and our environment. Exposure to chemicals like pesticides and herbicides, or especially glyphosate, which is used in a lot of GMO foods. Your hormones, as they fluctuate, and health status, such as treatment for medications or other medical interventions, and our age. Diseases and disorders being studied and being treated by microbiome test studies. As you can see, there's a huge list of diseases and disorders that they're trying to treat, and they have 331 clinical trials as of right now, seeing how fecal transplant from healthy individuals can help people with these certain disorders. These studies include helping treat gastrointestinal issues, mental disorders, and neurological disorders, both of which include the vagal nerve and the gut-brain axis, as we discussed earlier, metabolic diseases, inflammatory diseases, allergies, and immune response issues and infections. As you can see here, a healthy gut microbiome helps break down the food, provides vitamins and nutrients. It regulates our immune system, protects us from infection, regulates our metabolism, and plays a huge role in mental health. So how can we help our microbiome? Well, first, if you are interested in seeing what kind of diversity you have within your microbiome, microbiome testing is available. Ways to test your microbiome include home kits where you can send in a stool sample and the testing companies will send you the results. Another way is to have your primary health care giver give a test. This way your doctor can go over the results with you. Microbiome testing attempts to detect the presence of different species from microorganisms in a fecal sample. From the test results, you can get the information such as the richness of the gut microbiome, diversity of the gut microbiome, and how it compares to others. Consumption of probiotic-rich foods is very, very important. It's a natural way to build up your microbial environment within your body because they are naturally derived from these certain foods. So fermented foods such as kombucha, kefir, kimchi, sauerkraut, tempeh, miso, and yogurt are excellent sources of naturally occurring probiotics. Other ways to help is to consume fiber from organic fruits and vegetables. The bacteria in our stomach feeds off of fiber that we get from fruits and vegetables. So it's very, very important to feed your diversity of your microbiome. Consume less foods with added sugar. Try and reduce your stress. Limit screen time with electronics. Avoid taking unnecessary antibiotics. Exercise, getting enough sleep. Avoiding dangerous chemicals and cleaning products. Spending ample time outside, and definitely avoid smoking. Here's some additional reading for microbiome research. This one's really great. The human superorganism is really great. It goes back hundreds of years and how it has affected our genetics. So this is a really good one. The psychobiotic revolution is really, it talks a lot about mental health and our microbiome. And the microbiome solution is another one that helps explain it in a little bit more scientific terms. If you have any questions about the microbiome or you wanted to talk about it some more, feel free to email me here at my email address up here. And thank you so much for taking the time to watch this video.